All right, how y'all doing today? It's our Friday. All right, so let's turn off some of these front lights a little bit. So we've got to talk about Mendelian rules and with human genetics, we usually use pedigree analysis to look at inheritance patterns. So I want to talk a little bit about pedigree analysis. And now it just did it a second ago. Now it won't do it. So for pedigree analysis, you're relying on sort of tracking back a family tree, so to speak. You would have a person, maybe a patient who has a disease, or you might have parents that want to, they know in their family, maybe there is a disease that sort of runs in the family, and they're thinking about having kids, and so they want to maybe know or predict the potential for passing on a trait. So that's where pedigree analysis would come into play. And for pedigree analysis, there's a set of symbols that are used to create these pedigrees. That's sort of a shortcut for writing down bits of information. So these are the symbols that you'll find, and the majority of symbols you would find in pedigrees. Let's kind of walk through them briefly. You have symbols to indicate sex. Male, square, females are circles. And you have, there may be a case where you wouldn't know the sex, an unborn child or something like that, and that you could use a diamond for that. If you know the person is affected, the symbol would be colored in. Generally, you're only looking at one or two characteristics at the same time. That's about as much information as can be processed at a time. You can look at several things, but you're probably going to look at one or two at a time. So you only need to color them in and say, okay, they have the disease or they have that genotype. If they don't, you leave it open. If they're dead, you can cross them out, but that's not necessary. If we know that the gene is present on one of the sex chromosomes, you can use a dot, or you can fill in half of the box. Sometimes also, if the individual is heterozygous, we fill in half the box. But most of the time, neither of these two are going to be used. Okay, it's either just going to be affected or unaffected. In most of the cases, there's going to be one individual that was the initial patient or looking for the answer to the inheritance, and that individual would be called the propocytus or the propocyta, and they're sometimes indicated with an arrow. So you're like, okay, this is where we started, and then we went back as much as they could remember or find out about their family history of who had various diseases, and then they have children or they have brothers and sisters, and here we are going forward with generations after them. So with a family tree, you're thinking about generations, and the generations are going to be connected. So each mating between a man and woman that produces offspring, the man and the woman are going to be joined by a line, and then their offspring are going to be joined to that line by additional lines. And they're always listed in the order that they're born, okay? So first kid, second kid, third kid, doesn't matter if they're boys or girls. If you had twins, then what's the order? Who gets listed first? It's kind of irrelevant. If they're fraternal twins, they're connected by like an arrow or two lines making a triangle that come down to each individual, but the individuals are not connected. And if they're identical twins, they would have derived from the exact same genetic information. So basically, they would be treated as one individual genetically because they have exactly the same genotypes. And that's kind of like the kids are going to be put on two ends of a coat hanger. You could have identical triplets, right? So you'd have, you know, 3 people, but the last 2 would have a little arrow going down to them. And then notice that that comes up and goes to a single line, and the single line would go up to the parents. This is not used so much. It's not like you're like, oh, well, girls have the line and boys don't have the line. No, they shouldn't have done that. We don't want to do this because that's too easy to miss that right there, right? So you want to always have it come down and then split, and that's identical. If they're fraternal, there's no straight down. They both just split off. It just means they both happened at the same time, but they're totally separate. Okay, so fraternal twins are 2 different fertilization events at all. Also known as dizygotic, 2 offspring, versus monozygotic, 1 offspring that split at some point during embryonic development. Okay, there are instances where you're going to have consanguineous matings. That means 2 people that are closely related, brothers and sisters, mothers and sons, fathers and daughters, first cousins. Those would all be considered consanguineous because they are only 1 generation removed from one another, or not even 1 generation removed in the case of siblings. Okay? So as I mentioned earlier, you're using these types of things when you're wanting to maybe analyze a pattern in humans because you want to predict the possibility of having an affected child, or you want to track the inheritance pattern. And basically there's 2 aspects of the inheritance pattern that can be determined with pedigree analysis. The first is it inherited in a dominant manner or a recessive manner? Okay, so you can only determine dominant versus recessive. And so far, that's the only type of inheritance we've even talked about, but there are several other patterns. Okay, but those patterns are irrelevant to pedigree analysis. And you're going to basically be tracking a normal state or a mutant state. Okay, and so you'll have a normal allele. You're basically tracking a mutant allele. That mutant allele may act in a dominant fashion, or it might act in a recessive fashion. So the thing to keep in mind about dominant versus recessive, okay, how you're going to make that distinction. Dominant means 1 copy of that gene, or that version of the gene, 1 copy of that version will show up in the phenotype. It will dictate the phenotype. Okay? So you would have what would be considered the mutant phenotype if you carried 1 mutant allele. That's what dominant would mean in this instance. So recessive is when no dominant allele is present, okay? And you have what would be the recessive phenotype. So that might require having 2 recessive alleles, or even just 2 wild type alleles, right? So some diseases or some conditions might be considered dominant, right? In most cases, that means that that version of the gene has a gain of function. It functions in a way beyond the normal level of function or the normal position of function. It's functioning in a different place than it's supposed to, or in a different manner than it's supposed to. And it is going to show its actions where the normal wild type gene would just do the normal thing. So wild type would be recessive in that instance. The other aspect of the inheritance patterns that we can determine with pedigrees is sex linkage. Okay, sex linkage. Humans and all mammals, for that matter, sex is determined by chromosomal difference. You have an X and a Y for males, 2 X's for females. It really boils down to, do you have a Y chromosome? If you have a Y chromosome, it doesn't really matter how many Y chromosomes you have, and it also won't matter how many X chromosomes you have. If you have a Y and it's functional, you're male. Okay? You can have as many X's as you want, you're still going to be male. Okay? So sex linkage really is saying the gene that you're analyzing that controls the trait that you're analyzing, that gene is located on the X chromosome or the Y chromosome. Okay? Genes located on the Y chromosome may only have their phenotype affect males. So that's pretty obvious. Males will be affected, and all male children of an affected male will be affected. If that's the pattern you see, it's paternalistic and it's Y chromosome. Okay? On the X chromosome, it's a little more tricky because females having 2 X chromosomes, males have 1. So with X-linked traits, the mutant alleles show up in the phenotype in males more frequently than females, because for females to have a recessive trait would require both X chromosomes to be mutant, while as males only require 1 X chromosome be the mutant because they only have the 1 X chromosome. So an X-linked trait, sometimes it's called X-linked, sometimes it will just be called sex-linked because it's apparent it's not on the Y. So sometimes sex-linked can just mean X-linked, but it's probably more appropriate to say X-linked so that you're being as specific as possible. So an X-linked trait will show up more commonly in male members of the pedigree. So how would you look at a pedigree and then be able to determine which of those dominant or recessive, sex-linked or not? So I forgot to mention. So if it's sex-linked, right, that tells you it's on the X or the Y. What if it isn't on the X or the Y? Okay, so the word we need for that is autosomal. So that means it would be on any of the other 22 chromosomes, sorry, 22 chromosomes. So if you're looking at a pedigree, the first distinction that you'll try to make is this a recessive or dominant pattern? Okay, so there are a couple of clues that can help you. First off, if it's a recessive pattern, then you will find that you'll have phenotypically normal individuals will have mutant offspring. They'll have affected offspring. And it should be approximately 25% of the time. But that's only true if, and it can be untrue that that's the case, but it should be about that case. Now, how many people do you know, there's the exceptions out there, but how many people do you know who have 12 kids? Kind of rare, right? You know somebody, right? So one person out of all of us knows somebody like, so it's not common to have super large families this day and age. Most of the time, people will have 1 or 2 or 3, maybe 4 kids, right? So pedigree analysis is a little harder to do because you're limited with statistical predictions because you want to go, oh, well, 1 out of 4, but they only had 2 kids. So how do you get 1 out of 4 if you only had 2? So you can't really try to crunch the numbers that way, and you shouldn't from a pedigree. If you only have 3 or 4 kids, you shouldn't be trying to do a Punnett square with them and say, oh, well, 1 out of 4, right? You just need to look for sort of absolutes. You need to say, okay, if I've got 2 normal parents and they have any abnormal kids, then it's probably recessive because each parent probably had a mutant allele, and that one kid got both mutant alleles. However, if you have an affected individual, or sorry, two affected individuals, and they have kids, and it's autosomal recessive, then every kid should probably be affected. Okay, so if you see that, well, that's kind of a no-brainer. You're like, yeah, okay, well, two affected people better have all affected kids if it's recessive. Okay, if two affected people don't have all affected kids, that would be a strong indication that it's actually dominant. Okay, so a dominant pattern could be indicated when you have an affected individual, okay, who has an affected parent. Because if it was dominant in the parent and it gets passed on, it's going to be dominant in the kid, and their kids, they should have some affected kids. So with dominant genes, you usually see affected individuals in every generation. And the only sort of reason you might not is because mutations occur. They don't have to be passed down. That could be the first person, you know, who had the mutation. Maybe they're in the doctor's office and they're 40 years old, but when they were in their 20s, they thought it'd be a great thing to go visit the interior of a nuclear power plant, or not that that's necessarily dangerous, but say someplace like Chernobyl, right? So you're like really getting radiated, or maybe you got exposed to some toxic chemical or something like that, and it caused a mutation in that person. Well, now they have it, and they might be able to pass it on, but nobody in their history of their family tree has it. That's called a spontaneous mutation. That's the first instance of it in the family tree. And that's going to show up, but it may not look like, you know, that person may not have any kids yet. So how can you predict? And so really, to make predictions, you need both parent generations and offspring generations, okay, for pedigree analysis. All right, so let's look at some examples and then try to use some of these clues to deduce what might be going on. So here's a known instance for the disease cystic fibrosis. And you may already know about cystic fibrosis. Who's heard of cystic fibrosis before, right? CF. It is a disease that happens when a person is lacking both functional copies of the CTFR gene. And those genes can be altered in a variety of ways, and some of those ways are quite detrimental to functioning. Some completely eliminate functioning. Some just impair functioning, but there's some functioning left. And so you have a wide range of phenotypes for individuals with cystic fibrosis, from very severe phenotypes to mild phenotypes. Depends on how it is mutated. And if you have that disease, you have both copies of your gene are defective, okay? And so it would have to be recessive. So this is a family tree or pedigree of what a recessive trait would look like. And it's on an autosome. I don't remember which chromosome it's on, but it's on an autosome. So you have two unaffected parents at this generation, okay? And they have an affected offspring who has no children. So it's hard to make any real prediction from that. But if we look at his sister, she also inherited some of the same genes he did, right? Same parents, so same genes. Well, she has one child that is affected, okay? Now, both sexes are affected, in this case, in equal proportions. I would be very cautious to say if I only saw one of each to use the word equal proportions, because you haven't seen enough individuals, okay? That's just not statistically true to say at this point. But we can say that at least it doesn't preclude that both sexes can be affected, right? We don't know if one's more than the other, but at least both can be. So what are our clues that this would be recessive? If we didn't know already, the first clue would be affected kid from unaffected parent and another affected kid from unaffected parents. So the parents presumably would be carriers. An individual that carries a mutant allele or is a heterozygote is often sometimes referred to as a carrier. They carry a mutant, they carry a wild type. Okay, so that would be an example of an autosomal recessive. So a little bit about the symbolisms here. You have each generation in this tree written above the other. And so you've got your male and your female parents here connected by a line. They had kids. So these kids, this drops down and says, okay, all these kids on this line came from this mating. Okay, what would happen if they got divorced, she got remarried, and had some more kids? You would draw another line over here and have another male symbol and have those kids come down and be over here. Okay, so you would show them like that. When she got married again and had some more kids, you'd have another line that goes off. It'll go off and over and down. You'll just have it all spread out. But you want to keep each generation on the same row like that. So this is generation 1, generation 2, generation 3 in this family tree or this pedigree. Each member of the pedigree, regardless of their blood relationships, their parentage, or if they're just a member of that line due to marriage, they each get a number, right? So these 3 siblings, right? She got married, she got married. It doesn't matter which side of the symbol you draw, the male or the female, that's irrelevant. Okay, some people go, oh, you gotta put the square first. No, you don't, doesn't care, nobody cares. They're in order of birth, but only the siblings. This guy could be 14, and he could be 50, it doesn't matter, okay? But the siblings would be in order of birth order, right? So he's number 1 just because he's listed on the left. You're not number 1 because you're the first kid born or anything like that. You're just number 1 because you're the first person on that line in the second generation. So each individual then is going to have a unique number that they could be referred to by, right? 2-4 or 3-1, and you could tell, okay, that person, 3-4 is affected, and 2-3 is affected, and the 2 parents are not, and so on and so forth, okay? So without those symbols, it's really not a correct pedigree. So if you draw a pedigree, you want to throw those symbols in. Sometimes they will be written inside the box, sometimes above the box, sometimes beside the box, sometimes under the box. It doesn't matter where you write it, just so long as it's there and it's clear who got what number, okay? So now here's one I didn't tell you anything about. So can you look at that and let's make some predictions. Y'all make some predictions. Look at that for a few minutes and see what you think. What are some things that pop up? Shout them out.
Male and female are affected.
Okay, so both sexes can be affected, and it looks like there's 2 of each, so maybe it's kind of equal. No sex difference yet. So autosomal, right? If both sexes can be affected, possibly it's autosomal. Now, is it dominant or recessive? What do you look at there? You're looking at the already printed out version, you know. So what does it say? Recessive. Okay, so well, why is it recessive? Where are the 3 places you should look that recessive should pop up? Tell me what's missing. Well, yeah, go ahead and tell me what's missing from that pedigree that makes you telling me which individual is harder. Like if you drew that pedigree, what would I take points off for? First, give me the reference number for the first affected individual by looking at the screen. Huh? I don't see a 1-1 up there. Where do you see 1-1? It's not there. That's what I'm trying to get at. That's missing. That makes it really hard to just say, okay, well, 3- whatever is affected, but her parents are not. Okay? So I messed up. I left off the, I don't think it comes up when I click the button either. But if we're going to say it's recessive, we can start filling in some info. Like, okay, if they're affected, they must have a homozygous recessive phenotype if we say it's recessive. So we could say that their genotypes would be homozygous recessive. We'll just use little a for whatever, for anything. We don't care what it is. So what can we tell from them about their parents? If you have an unaffected individual, what is their genotype going to be? How much of their genotype do you know? They're either heterozygous or homozygous dominant. So we know that their genotype has two possibilities, actually, right? A something, right? We can use the dash. We don't know what their genotype is. So like for this, the original parent up here in this generation, she's probably heterozygous because we know at least because she's not affected, okay? And we know that these parents are not affected, so they must have been heterozygous. But we also know that they had an affected kid, so that's what tells us that they're heterozygous. They must have had a dominant allele because they're normal, but they must have had a mutant allele because they made a mutant kid, okay? Well, that tells us that and that and that. So if she's heterozygous, we know that she got that little a from dad and the big A from mom. And every one of these would have had to get their big A from mom. What's the likelihood of getting 4 kids that all get a big A if half the time, if she was heterozygous, half the time they might have gotten a little a, right? Nobody did. So that makes it seem more likely that she was probably homozygous. Okay, now we can't say that with 100% certainty, but we can say what the probability would be of getting 4 kids if she was heterozygous, and say that that probability is too low, and we can then presume homozygous. Okay, so you can learn some probability things from a pedigree, and that's the next thing we're going to talk about is probability. So I'm going to kind of throw it in there. Okay, what's the likelihood? You can say, well, what's the likelihood of getting that many people with that genotype from parents if the parents they were heterozygotes or if they were homozygotes, whatever. Okay, here's another one. Okay, so what pops out at you from this pedigree? Does it affect males more? 4 males, 8 females. Yes. We've done a news media analysis of the data. Okay, so who's actually affected more? Females. Okay. But both sexes are affected, so sex linked might not be the first thing to think about. Actually, linkage might not ever be the first thing to think about. The first thing to think about would be trying to say is it dominant or recessive. Are there any affected individuals whose parents were unaffected? Are there any affected individuals whose parents you don't know about? There's always some missing information in a pedigree, right? So when they got to this point, you probably go, let's find this dude if he's still alive and get some pedigree information from him. I mean, you can do that. You can add two pedigrees together. So let's find out sort of what's happening there. Was one of his parents affected, or were neither of his parents affected? Okay, that might give you some information that's missing right here. But if we have to go based just what's here, does anyone have an unaffected? Does anyone have both parents who are unaffected? Both parents. So one is affected. Parents, one is affected. One affected. They didn't have any kids, so they're uninformative for us. He has kids. He's affected. They're affected. They're unaffected, but sadly, they didn't have any kids. All nerds. So here, we got some more kids. So every time we have an affected parent, we also see that they're making affected children. So that follows one of those clues. Which clue? Recessive or dominant? Much louder and much more clearly. Dominant. It does sort of seem like it might be dominant. Every time you have an affected parent, you have affected kid. Now let's look at what sex the affected children are with respect to the affected parent. When you have affected females, what sex affected children are present? So let's find an affected female who had kids. It would have been so much helpful if I had put the symbols in, right? We would have said, oh, 3-5. 3-5. She had affected children. Did she have both males and females? Yes. Okay, so females can pass it on to both sexes. Okay, we know that so far. Let's look at affected males. Here's an affected male. Did he have one sex or the other affected, or both sexes affected? Only females, but he only had 3, so don't jump to a conclusion, just say, correlates. How about this guy? Only the female. How about this guy? Now here's the money guy. He had 5 kids. All 3 girls were affected. That 3, and none of the males. So if you think of each generation, look at a generation with an affected male, okay? This generation has affected males. And if we look at that generation, all affected males have only affected daughters. And if we look up here, that affected male had only affected daughters. So now there's 3 males who are affected, and all of their daughters are affected. Amongst them, none of them had an unaffected daughter. Okay? That's now a stronger argument to say, okay, something's going on here where this trait is being passed on perhaps differently from the fathers than it is from the mothers. Okay, why would that be? Well, the fathers have an X and a Y, and the mothers have 2 X's. So this gets us back to thinking possibly sex link. We can't think sex link because more males are affected than females, because that's not even true, is it, from this pedigree? But we can go back to thinking sex link because we look at the parent and the sex of the affected individuals that they have. So the females have both, but the males have only female affected females. And so there's a difference between the 2 parent sexes based in regard to the sex of the affected children. So males, we can think about their genotypes. Come on, and pop up. Males have an X and a Y. Females have 2 X's. She's not affected. We've decided this might be a dominant trait, so therefore she must not have that dominant trait on either of her X chromosomes. So we could fill in X plus, just normal X. Plus just usually means normal. And then you put something else, like a minus symbol or an allele symbol or something to indicate that's mutant. He's got a mutant X. So we'll put it, and it's dominant, so we'll put a D there. And his Y. So when he passes on, every daughter gets one of their X's from their father. Okay? Every female has 2 X's. One of them came from dad. And dad only had 1 to pass on. So you and all your sisters have the one same X chromosome that your dad gave you. Okay? You girls have sisters? Which girls have sisters? Okay, your sister and you have 1 X chromosome that's identical. All right? So, boom, affected, because it's a dominant trait. Doesn't matter what the mom gave him because the mom's chromosome is not controlling phenotype. But what about Bubba? Well, he got his only X from mom, and mom's got good X's, so he's fine. Okay? Now she has kids. Well, she passes that X on 50-50. All of her sons get one of her X's, but it's a 50-50 chance to whether they get the dominant mutant X or the wild type X. So some sons are affected, some sons are not. And they drew this pedigree to give you perfect 50-50. 2 sons are affected, 2 sons are not affected. But also, one of their daughter's X's came from the mom. So it's a 50-50 chance for the daughter to be getting the mutant X. This one got it, that one didn't, this one got it. Okay? Now, when he has that mutant X, every one of his daughters are going to be affected. Every one of his daughters are going to be affected, and none of his sons. So this points to what we would see as an X-linked dominant trait. Okay? X-linked dominant. Males will pass on that trait only to females. So if you see only daughters are affected of affected fathers, that's kind of a clue to sex-linked dominant. All right, one last one. What's going on here in this unlabeled diagram, unlabeled pedigree? First go dominant or recessive. Recessive, because, boom, you got unaffected parents having affected kids 3 times. Okay, here, those 2 guys are affected. Here, their kids affected, right? So 3 times you got affected kids from unaffected parents. That really says recessive is probably what we're going with. Now, is it on the X chromosome or the Y chromosome? Is one sex affected more than the other in this instance? Yeah, so it's like all to none, right? Only males seem to be affected. So that would make us think recessive, sex-linked, recessive is probably what we would say. Could we fill that in? Could we plot out genotypes that would support that? Okay, so if it's sex-linked recessive, then these males must have a mutant X chromosome. I just indicate it with a minus sign, so say something's wrong. We don't know what trait that is. Maybe let's say it's color blindness. That's one that goes on the X chromosome. So, it could, or hemophilia, or fragile X, and who knows. So they're going to have it, and these guys down here must have that genotype as well. So what can that tell us about their parents' possible genotypes? Well, this male was normal, so we can go ahead and throw a normal X on him. And the mother didn't have it, but some of her kids did. So she must be a carrier at least, right? But we can actually say she was heterozygous because if she had had 2 mutant recessive alleles, she would also have been affected, okay? And so that can happen. When you say, oh, sex-linked recessive doesn't happen in females, of course it does. If that person has 2 mutant X chromosomes, they'll have that. So are there colorblind women? Of course there are, okay? Are there more colorblind men than women? Yes, because you have to have 2 mutant copies to be colorblind if you're female. All right, so she's heterozygous, right? And her mutant X has been handed off to these boys, but not this boy down here. He got the normal X, okay? And of course, her daughters got a normal X from dad. So even if they got the mutant X from mom, they're going to be not affected, right? But these two daughters here had affected sons, so they must also be heterozygous. So we can go back and say, okay, well, now we can actually probably figure that their genotype is heterozygous. And what about this son down here? Since he's not affected, we can say he's also X plus Y. What about this daughter? Can we say anything definitive about her? If yes, what? And if not, why? Correct. She did not have any offspring, so we can't really make a good conclusion about what her real genotype might be, but we could say we know at least she's got a normal X. We don't know about her other X. One X could be, she could be homozygous normal, or she could be heterozygous. We would have to see what happens. We have to say, okay, get on Tinder and let's get some kids here. All right. Is that what that's not what Tinder's for, is it? Oh, it is? Okay, okay. I'm trying to keep up with the cool new stuff. So that's kind of always the thing about pedigrees is if individuals didn't have children, it's, what can you say? You can't really know what they could have passed on, or you can't know what they have because you don't know what they passed on. All right, so that kind of wraps up. So pedigrees, your book has a few problems on them you can look at. I don't remember if I assigned any in the online homework or not, but you might come across them at some point. And the thing about it is, if you're just tracking back for a disease, you're kind of just looking for who might have had it, right? The real reason, so medical doctors would kind of look at pedigrees now is they'd say, okay, we could go and look at your sister or look at your aunt or your uncle or maybe your grandparent and get some DNA samples and see if we can track a potential a mutant allele. Like if you had a disease and they don't know what's causing it, they could actually try to find that gene by knowing that these individuals all have it and these individuals don't, and then they could compare and contrast their genomes through DNA sequencing to see, okay, what's different? And then look at the differences, and those become candidates for what might have gone wrong, right? That's kind of the approach there. The other use, really heavy use for pedigrees would be predictive. Like, okay, she wants to have kids. She's like, my brother's colorblind. Are my kids going to be colorblind? If they are, what's the likelihood that they will be? Can I look at my future husband's or sperm donor's pedigree and see if he had any colorblind people in his pedigree, to see if he might carry that mutant allele, and then what's the probability, if he does, that I might have kids, or what's the probability that if he doesn't, that my kids won't. So it might be a predictive thing, and that means you need to understand some probabilities. So that sort of is where we want to go now and talk about probabilities and statistics. Okay. Fun little book I like. It's called Damn Lies and Statistics. So the thought is, if you can apply statistics to something, you can make the numbers say whatever you want them to say. You can misinterpret them in lots and lots of ways, and persons who are you're telling to, without the necessary training or time to really analyze what they're being told, will take your word for it, because if you said probability 100%, and they're like, oh shit, and they're like, yeah. So what's the probability of you dying? 100%. Are you scared now? Exactly. So it's all just a big lie. All right, so what we do with probabilities of Thought I already did that. What's the probability of certain things? And so breeders are going to be concerned with this because they're wanting to know sort of what's the likelihood of inheriting certain traits and making a better crop or making a better animal of some kind, right? Making a pot-bellied pig. Parents would want to know, you know, making family decisions or having kids. So the probability, pretty simply, is the likelihood of an event happening. And you determine it by saying, okay, how many total number of events were there and how many events happened X or Y, whatever event that you're talking about, right? Like flipping a coin. How many times do you get heads out of how many times do you flip? Each time you flip, you have only two choices, heads or tails. So to get a head is probability of 1/2. To get a tail is also probability of 1/2. They also make it something a little more tricky. What if you had a 4-sided Dungeons and Dragons dice? You're like, what? What? No, don't worry about it. You had a 4-sided dice, a tetrahedron. What's the probability of coming up any one of those 4 sides? 1/4, right? How about a regular die from Monopoly? 1/6, right? So it's the number of times any particular thing could happen out of the total number of times or total number of possible things that could happen. Another thing about probabilities, and this is sort of leads into the trickery that people can play with probabilities, is to think about sample size, okay? Large sample size, less error. Small sample size, more error, okay? So if you flipped the coin 10 times and you got 7 heads and 3 tails, you wouldn't really think anything of it. You wouldn't go, oh, you're cheating. But if you did it 1,000 times and came up 700 of 1 to 300 of a 1, well, 700 is a much bigger number than 7, right? Magnitude. So you're thinking, shouldn't it have been more like 450 and 550 or 499 and 501 or 500 and 500? Shouldn't have been closer to half, right? So yes, it should have. And so if you're off, you know, by 70 to 30 and you did it 1,000 times, okay, call the dude over. They're cheating the dice, okay? So for doing good statistical analysis, you need large numbers of things to analyze. So when we do that in genetics, it means that's why we said it's harder to do with something like human families because you don't have 75 kids to look at. But you could do it with fruit flies because they're going to produce thousands of offspring, or at least hundreds. And so with organisms that produce a lot of progeny, statistics are more applicable to those studies. But we have statistical methods that allow us to study small sample sizes with levels of confidence that allow us to make sort of decisions about it. But the statistics that we're typically going to be looking at are predictive things, okay? So we have essentially 3 different mathematical operations that we can consider doing. 2 of them are really the same thing. One is a simplified version of the more complicated version, and the other one is a different thing altogether. So the first 2 here, the product rule and what's called binomial expansion are the same. The product rule is a specific case of binomial expansion when order of occurrence is taken into effect or into account. And binomial expansion would be a situation where order of occurrence is irrelevant. So the product rule would be used when you're saying, what's the likelihood of this happening and then this happening? Okay, in order. And the binomial expansion rule will be say, what's the likelihood of this thing happening out of 5,000 things? We don't care what order. It can come last, it can come first, it can come in the middle. So order doesn't matter. And the sum rule, So product means multiply, and the sum rule means add. And the sum rule is used for events that are mutually exclusive, right? Like I said, what's the probability of dying? I said 100%. What's the probability of being alive or dead? Also 100%. But can you be alive and dead? I don't mean some physical meta awareness thing, like I'm alive, but I'm dead inside. I don't mean that. I mean, can you be breathing and not be breathing at the same time? No, right? So two mutually exclusive events can be calculated. You have to use the sum rule to do that. So the thing I always think about when I look at a situation where I'm being asked about these is, am I able to phrase it in some way to say, what's the probability of this and that? If I can throw an and in there for the two or three things like this and that and that, if you can put an and in the description, product rule. Okay, two events happening simultaneously, product rule. Okay, so what's the probability of being alive and married? Right? You can be alive and not married. You can be alive and married. Dead and not married. You can be. Oh, okay. So anyway, alive and right, two not mutually exclusive things. So you would use the product rule for that. Two mutually exclusive things, you have to use the sum rule. And we'll look at some examples. So we already have used the term independent assortment, right? So Mendelian, we've already used the product rule when we talked about the independent probability of being a phenotype like tall and yellow for the P's, right? So two independent phenotypes, we would calculate the occurrence of that using the product rule. And that's what we did when we did our fork line method. We said there's a 3/4 probability of being yellow, there's a 3/4 probability of being tall. So what's the probability of being yellow and tall? 3/4 times 3/4, right? One event, the occurrence of one event has no effect on the ability of the other event to occur. Let's say we've got something like another example, a human trait, congenital analgesia. You don't feel pain. Maybe it would be good, maybe it would be bad. You get burned or something because you don't realize you're getting burned, that kind of thing. But anyway, we'll give a couple of allele symbols for it and say, okay, well, you've got a couple of heterozygous people. They're going to have kids. What's the probability that their kids, they're going to have 3 kids, what's the probability that those kids, which we could also say the first 3 kids, it's the only 3 kids, but what's the probability of their first 3 kids would have that phenotype, right? Well, they're going to have to inherit 2 copies of the mutant allele to do that. And we can use the Mendelian, like a Punnett square, or our known Mendelian phenotype or genotype ratios to know what the probability is. So the probabilities in genetics that we're getting are those probabilities of the gamete formation. So what are the individual probabilities? As we said, if we crossed 2 heterozygous individuals, we know that for the genotypes, there's a 1/4 chance of being homozygous dominant, 2/4 or 1/2 chance of being heterozygous, and 1/4 chance of being homozygous recessive. And the question is asking really about these individuals, the chances of being homozygous recessive and having the phenotype of congenital analgesia. So the probability that we're really going to concern ourselves with is a 1/4 chance of having that versus a 3/4 chance of not having it, right? And we do try to put our probabilities into 2 possibles, right? This or that, right? So the probability you're asking about of that occurrence for any individual would be 1/4. Okay, so for any kid, the probability is 1/4. Now you have to think about, well, if the first kid had that, inherited that genotype, would that in any way alter the probability of another child inheriting that genotype? And the answer is no, okay? Each meiotic event is independent of all other meiotic events. So one kid is the result of a meiotic event. The next kid is also the result of a totally independent meiotic event, okay? You don't have any sort of knowledge that when one oocyte or one sperm has been made with a particular, carrying a particular allele, that another sperm or egg is being made can in any way alter what alleles it gets based on what alleles that one got and what alleles got passed on to a kid, right? The next oocyte has no way of knowing, oh, whoa, 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 whoa. We already got a kid with that allele. Throw that one away. We need that allele. No, that's not going on. There's no conscious decision in oogenesis or spermatogenesis. They're just random, okay? So the probability is the same every single time that meiosis occurs, that any of those possible gametes could be made and any of those possible genotypes could occur after fertilization. So every kid has exactly the same probability for that genotype. So the first one has got a quarter, the second one's got a quarter, the third one's got a quarter, but the question said, what's the likelihood of all three of them having it? Well, since it's independent, we go, it's the probability for child 1 times the probability for child 2 times the probability for child 3. So the actual probability of all three in a row is 1/64th. So it's pretty low that all three kids are going. Well, what's the probability that the first two kids have it? 1/16. What's the probability that the first and the last kid have it? What's the probability that the second and the last kid have it? Right? What's the probability that 2 out of 3 kids have it? We're going to say 1/16, but that's not true. Okay? Because there's 3 different ways for 2 kids to have it. So the probability actually goes up. One way for the 2 kids to have it is the first 2. Another way is the first and the last. And a third way is the last 2. So there's 3 ways for 2 kids to get it. So the probability is actually not 1/16. It's 3 times 1/16. Which would be 3 something. Anyway, so we can leave it as 1/64th. We can convert that to a decimal, which is what's normally done, because when we talk about probabilities, we don't usually talk in terms of fractions. You don't say you have a 1/10 of a chance, right? You don't write 0.1 or 1/10. You would say you have 10% or 1% or 0.1%. You put percent at the end of it. So you'd have to turn that into a decimal equivalent, then multiply by 100, and that gives you the word percent you can put there, or the symbol percent. So not a high probability that first 3 kids. Now, how would we actually go about calculating that more complicated question I asked about what's the probability of 2 out of the 3? Any 2. We would need the binomial expansion to do that. And we're going to jump to that in a minute. It's a little bit tricky. Okay? We can look at probabilities for phenotypes, but we could, of course, that's because of this particular genotype. So really, you're looking at the probability of getting a genotype. So we could just look at the probability of a genotype. Here's another example. 2 traits for some mouse, ears and tails. We don't care. We got droopy ears or crinkly tails, whatever. Right? And so a dihybrid cross, we've already looked at this. How would we work this out? We would use a fork diagram. Okay? So the probability of getting this genotype and that genotype. Okay? Notice what I said? And. Right? You can have both things at the same time. Okay? They're independent. So what's the probability of being dominant homozygous for the droopies and dominant homozygous for crinkly. It's those two probabilities multiplied together, so it's 1/16. That's the probability of being homozygous for both traits. What's the probability of being homozygous for droopy and heterozygous for crinkly? What's that one? Okay. So each one of those, each aspect of the ratio of a dihybrid ratio, 1:1, 2:4:2, 1:1, is a probability. Okay. 1:1, 2:4:2, 1:1 is a proportion. 1/4, 1/16, 2/16, 1/16, 2/16, 4/16 is a probability or a fraction. Okay, so when you think about it like that, that's also the probability. And you can do it, of course, for all of them, right? Fork line method. And I like this slide because this is how I do fork line methods. I don't write this down. So I don't write these three things down for this one and then write those three things down again, write those three things down again. That's extra time that I don't want to take when I'm doing something with these. I can draw lots of lines much faster than I can draw or write those things. So I just make lines. If you have 3 different colors or 5 different colors or however many different colors you need of pens, that's helpful. Otherwise, you just gotta focus. Don't lose track. Okay. So we did it. This is the same thing, except I filled in the genotypes, but it's the same thing. So the fork line method is nothing other than the probability rule used for genotypes or phenotypes if you want to turn the genotype into a phenotype. There it is again for 3 genes, okay? All right, so what if 2 things cannot happen at the same time? They are mutually exclusive, okay? How are you going to know? It's pretty obvious deductive reasoning. Can you have a crinkly tail and a straight tail at the same time? No, you don't have 2 tails, usually. So that would be a mouse. Is it? Tale of 2 cities? Tale of 2 tails? Well, if they're mutually exclusive, you're going to be able to word the problem where you would say this or that. Or. You can't be both. It's one or the other. That's going to say, all right, I know I need to do the the sum rule now. So we'll use our same mice. All right, we know that for the phenotypes to be normal and for both traits would be 9/16. 3/16 would be just crinkly tails. 3/16 would have just droopy ears, and 1/16 would have droopy ears and crinkly tails. So you never have a chance where you have droopy ears and normal ears at the same time, right? So these are completely normal, and these are completely mutant, right? So you might want to think about what's the probability of being one way or the other. So let's ask the question, like, what's the probability that an offspring would be either completely mutant or completely normal, right? So they're either this phenotype or that phenotype. How do you calculate that? Well, all you have to do is add the probabilities of those 2 individual events. Add them. Where we already know it's 9/16 to be normal and 1/16 to be double mutant. So you just add them together. The probability of being this or that is 10/16 or 62.5%, right? Let's say you had a mouse raising facility and you knew people would buy cute little mice. And you also knew that they're really adorable if they have the little puppy dog ears, because their ears are droopy and they have a little crinkly tail, right? They're like, oh, aren't they cute? And so you know people are going to buy those. So you could sell those mice for $5 a mouse. And the other mice, they're not going to be so desirable. People are going to buy them for like snake food or something, feed their falcons. But they're not going to want to pay a premium of $5 a mouse for their snake. They're like, yeah, I'll just turn the snake loose and let him fend for himself. So they might pay $1 for a mouse or 50 cents or something, right? So you're trying to calculate your profit margins. And you're like, okay, if I have X number of mice and they have this many babies and I've got to feed them this much a day and I'm going to make this much on them for this kind of mouse and this much for that kind of mouse, then the X amount goes this and this, right? So you think about being a geneticist and being an entrepreneur requires a little math, a little knowledge of statistics. And you're going to be like, okay, I got to have this many mice and I got to sell them for this price to get this return. If they're going to have crinkly tails and I've got 10/16 of them that I can sell for $5, that means I've got 6/16 of them that I can only sell for $1. So I've got to have this many of those to offset this many, right? So there might be some ridiculous reason why you'd want to know that probability. I'm just making stuff up. Like, why would you care? But that's the sum rule, okay? You'll hardly use the sum rule. You will lots of times use the product rule because of Mendel's second law, which states, this is where you just regurgitate it right back to me that quick. Mendel's second law. Nobody wants a gold star for today. Why did I tell you to memorize that? That's the name of it, but what does it say? Okay, pretty much. Either allele at one locus may go together with either allele of a second unlinked locus. Hopefully you wrote that down in your notes, because I told you to write it down in your notes, and I told you to memorize it, because I would ask you, and you're like, nah, he's not going to ask us. Bullshit. You might want to know, what's the probability of a pure breeding organism? What makes something pure breeding? Why would an organism be considered pure breeding? Okay, so mating that organism or its organisms of that phenotype, that genotype would produce the same exact phenotype over and over and over again. Why was the question? Why? Because it's homozygous. So if the genotype is homozygous, it would be considered pure breeding. So what's the probability of a homozygous genotype from a cross of two heterozygotes? There's the cross, heterozygote, cross with a heterozygote. There are the genotypes. What's the probability of being homozygous? 1/16, right? No. 2/16, right? No. 3/16. Not lucky. Third time wasn't a charm. One more time. 4/16. Homozygous, 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 homozygous. No, they're not homozygous. Yes, they are. Do they have two little d's? Yep. Do they have two big C's? Yep. They're homozygous. Doesn't mean dominant and recessive. I didn't say no dominant and recessive shit. What are y'all listening to? I said homozygous. And y'all were like, oh, it means dominant. No, it doesn't. It means for each allele, for each locus, are the same. So when you have two bigs and two bigs, that's homozygous. When you have two littles and two littles, that's homozygous. But it also means if you had two bigs and two littles for different loci, that's homozygous. So the probability is 4/16, and you got that by adding each of these, because you can't have this genotype and a second genotype at the same time. That won't work. Each organism can only have one genotype, unless they're a chimera, and that we're not talking about that, okay? All right, so let's finish off with the complicated thing so it sticks in your mind. Binomial expansion. The binomial expansion is a way to calculate using the product rule approach. The product rule is a simplified case of binomial expansion when only a certain order can happen or the order is known. But when the order is unknown or unnecessary to be known, the binomial expansion can still give you the probability of those events. So you have to narrow your events that you're asking about to two outcomes, two possible outcomes, right? So if you said, what's the probability of having blue eyes? Okay. Are you going to worry about the probability of having brown eyes? Green eyes? Hazel? So the probability is blue eyes or not blue eyes. Okay, you take whatever it is and then you put not in front of it. Those are your two choices, right? So here are what all those things stand for. Probability is the probability that you've been asked about in the question or that you're assigning for a subpart of the question. N is the number of events. Usually it's the number of offspring you're being asked about, okay? X are the events that are of the outcome that is in question. It's the thing you're interested in. The blue eyes, okay, in our example. N minus X are all the other options, all the other different eye colors, for example. P and Q. P is the probability for the genotype or the phenotype that you're considering, okay? And Q would be the probability of all other things. So P plus Q must equal to 1. All the probabilities that we will be used in any sort of probability calculation are a decimal value or a fractional value. They're less than 1. There is no table. That triangle, don't ever try to use that. That's the triangle, no. No. Because you can memorize this and you have to redraw the table every time. You can use whatever you want. Let me back up. You're grown people. You can use whatever method you want. Now ask yourself, what method can I use that will answer a question in 5 minutes? Maybe drawing a binomial expansion table is not the answer, or a Punnett square is not the answer, not the approach, I should say. That's why I said that. All right, so the probability needs to equal to 1. And when you do this, this part of the term right here, as she was alluding to, you could use a table to find that term. That term is essentially how many different ways, how many different possible ways that outcome could be obtained. The number of different possible ways. So in our little example a while ago, 2 out of 3 kids. How many ways were there to get 2 out of 3 kids? Right? You could have 1 and 2, 2 and 3, 1 and 3. There was 3 ways to get 2 things out of 3 kids. How many ways are there to get 2 things out of 4? 1, 2, 1, 3, 1, 4, 2, 3, 2, 4, 3, 4. 6 ones, right? I'll do them all. And order doesn't matter. 4, 3 is not different than 3, 4. All right? To get that, you use factorial. And if you haven't had algebra ever, factorial means you multiply all of the integers, including that integer, down to 1, multiply them together. So 0 factorial is been defined as to be 1. 1 factorial is also 1. 2 factorial will be 1 times 2, so it's 2. 3 factorial is 1 times 2 times 3. 4 factorial is 1 times 2 times 3 times 4. 24, and so on and so forth, right? So what's 8 factorial? Don't give me the number. Tell me what it is. You can always not have to do the actual factorial because you're always going to be able to cancel something. You'll see in a minute. But it's 1 times 2 times 3 times 4 times 5 times 6 times 7 times 8. And if you know that, you can cancel stuff and you don't have to do the calculation. You'll see. Second. All right, so let's say we have a couple of heterozygous brown-eyed people and they're going to have kids, and if they have 5 kids, they might want to know, well, what's the probability that 2 of our kids will turn up with those ugly blue eyes? Because they know they're heterozygous. So what's the probability for having the blue eyes? Of course, you can work that out. You got 2 heterozygotes. The probability of being homozygous recessive is 1/4. So the probability of not being that is 3/4. And you could put, the only option was brown in this case, but you could put not blue eyes right there. So the probability, and then that thing that you're asking about, put it down as a subscript. You don't have to use parentheses. You don't have to write it out, but use some symbol that lets you remember. Okay, so N is the total number of events, which of course is the number of kids, so that's 5. How many do you care about? 2. You want to know what's the probability of 2 of the little suckers having blue eyes. So you go 2 for X. So N minus X is 3, right? Substitute it all in. There's your formula, which is easy to memorize. Plus, I always give it to you on the test or any quiz or anything like that, so you don't have to memorize it, except know where to plug stuff in. So you plug in your 5 factorial, or 2 that you're asking about, 3 factorial here, or 5 minus 2. What's the probability? P is the thing you care about. It's the thing that's in the question. The blue eyes in this case. So that's 1/4. X, how many of them are there? 2. So you raise it to the 2nd power. 3/4 is Q. How many are that? 3. Raise it to the third power. Now here's where I said, don't punch the factorial key on your calculator yet. That's wasted punching. First, use your biological calculator to go, oh, that's 5, 4, 3, 2, 1. That's 3, 2, 1. 3, 2, 1 cancels 3, 2, 1. I don't need to do 5 factorial. I just need to know that it's 20 divided by 2. You can do that in your head, can't you? Right? If I said, here's a $20 bill, give me 2 bills, what are you going to give me? I hope you're going to give me 2 $20 bills. So I'm be like, yeah, and here's another 20, give me 2, yeah, and I can make some money. But hopefully you know how to do money math. So if you calculate this out, you'd have 10 times 1/16 times 27/64, which is 26%. Okay, so once you've simplified your formula, now punch it into your calculator. Okay. And this is where I tell people, you can have the fancy calculators. If you know how to use them, that's fine. You're never going to need to do any graphing or any of that stuff. So all you really need for us is a simple calculator that would let you do like a square or a square root, maybe, if you wanted to do that. So what was the probability for 1 kid? If we said, what's the probability that child number 1 has blue eyes? What would you say? Huh? Talk louder. Guess louder. 1/4. Correct. What's the probability that the second kid, not the first one, not the third one, the second kid has blue eyes? 1/4. Or the third kid. 1/4. Right? What's the probability that the first kid and the second kid and the third kid have blue eyes? Oh, no, we only care about 2. The first and the second, what's the probability? 1/16. That's bigger than 1/16. Right? That's bigger than 1/4. Why? Well, because there's 10 different ways that it could happen. And any way is equally likely. 10 different ways to put 5 things together. Right? Let's see if that's true. One, 2, 3, 4, 5, 6, 7, 8, 9, 10. Holy shit, it's true. There are 10 different ways to do 5 things. Now, if you know what row to look on your table, you can get 5 or 10 or whatever. Okay? If you also know what to plug into the formula, you can get it without having to guess. All right, so the binomial expansion can be used that way, but a more common way is actually harder. So what if they asked the question, not what's the probability that 2 of our children, what if they said, what's the probability that at least 2 of our children have blue eyes? At least 2. Is that just one thing? 2 children is one thing, 2 kids. There's 10 different ways to get those. What if they said at least 2? That's not one thing anymore. You cannot calculate one probability anymore. You now have to calculate a probability for every different possible thing that's happening. Okay? So this would be if 2 had it, but is 2 the only answer, right? At least 2 out of 5. So if you had 1 kid with blue eyes, would that satisfy that request? No. If you had 2 kids, would that satisfy? Yes. If you had 3 kids, would that satisfy? Yes. 4 kids? Yes. All 5 kids? So there's 1, 2, 3, 4 ways to do it. So now you got to calculate a binomial expansion for 2 kids, do it again for 3 kids, do it again for 4 kids, do it again for 5 kids. But if you had 5 kids, that was mutually exclusive from having 3 kids. So you calculate each binomial and then you add those binomials together. Or you could turn the question around and say, what's the probability of not having the brown eye or the blue eye kid, right? And then you only figure it out for none and 1. So you only have to calculate 2 things. All right, we'll do that on Tuesday. So have a good weekend. And remind me that we stop at 9:20, not 9:30. I'm getting off. That's why I've gone over these past 2 times. I apologize.